Okay, so I'm very happy that I got this invitation, although I need to confess that I don't do too much plant research anymore. I did in the past a little bit, and now we do mostly clinical research applications, but I want to basically we use the same instruments, right? So I want to show what we do. What can we do some with mass spectrometers. So for those who doesn't know where is Vancouver, is in this part of the Pacific Northwest. It's a really beautiful city, and it's one of the reasons that I really am happy working here now. And this is the campus. The UBC knows this place pretty well, and we are very lucky to be in this place and be able to do some research. And this is the Life Science Institute. The mass spectrometer lab is in this part of the building. And this is a basically new lab that we have some instruments basically from Dr. Leonard Foster. And this lab is brand new. We started about 2 years ago, and that's the reason I got recruited here in the UBC. And we have a high resolution mass spectrometers. I will talk a little bit of some applications that we did in the past, not with this instrument, but similar. And actually, some of them were with this timsTOF Pro. Basically, this is the configuration that we use for mass spectrometry. We always have a UHPLC with a mass spec with different capabilities. These are time of flight, so they are high resolution. Here we have another HPLC is behind UHPLC actually, and we have another one. We have 3 high resolution mass spec, and another one is coming in this fall. So I'm gonna talk a little bit 6 applications that I worked a little bit in the past, and this the first thing that I'm gonna start is with the building capabilities for plant scientists, and we have a library that we acquired here, an in-house library. Then I'm gonna talk a little bit about some using the same analytical run to do both quantification of some compounds and at the same time doing targeted for profiling and fingerprinting some plants or products, extracts. Another one is a recent publication that we did using a mass spectrometry in combination with bioassays to find bioactives in some cellular models. A little bit about how we can use mass defect analysis to look for specific families of classes of compounds. A case that we worked that was pretty interesting, working with somatic embryogenesis to find some factors that they produce embryonic repression in coffee. And some we work a lot with honey in this lab, so we are working in determination of floral biomarkers. So basically, we are trying to see if the honey that we are buying in the market is the flower that they say it is. And a little bit of molecular networking that you have some talks in the past here. So I'm gonna go very brief in most of them. I just have a few slides just to give you the idea what we can do with these mass spectrometers, and then hopefully we can have a discussion for your ideas and how can we help in the future. So one of the first things that we did is to have a in-house library. You know the difference between having an in-house library and external library like HMDB, Knapsack, Medlin, is that we can, when we have that compound and we run in exactly the identical conditions that we run your plant extract, we can call those level 1 annotations. And those annotations are more precious than the level 2. Because level 2, basically, we know is very likely that it is. But unfortunately, in metabolomics, we don't have a false discovery ratio control as in proteomics. So it's a lot more complicated to sell level 2 annotations. But for that, we use software. And this window is a software that is called Progenesis QI that we can put inside our in-house library, but also many other libraries. As I was mentioning, probably for plant research, the very interesting ones could be like Knapsack or Phenol Explorer, or even Medlin has a lot of products that are natural products. Cake also, and even human metabolomics database, even though it seems like it's for human, but many of the metabolites in humans are coming from our food, and most of that could be from plants. So okay, let's start with the first application. As I was mentioning, this is we have the option when we do mass spectrometry to go 2 pathways. Basically, we can go for non-targeted approach that we call untargeted metabolomics. And for this, we basically acquire as many data points as we can from the data, and to then screen that against libraries, okay? And the other one is targeted. When we know some of the biomarkers of our plant, like the compounds that we think those are the bioactive, then we can look for getting a standard of those and do all the validation of our method to quantify them. This work, and the reason we can do both things is because there is different ways to acquire the data, and one of them is called data-dependent acquisition. So basically, this is what is happening in the instrument. When we have, imagine that the ions are coming out of the column and they have different size and different abundance, sorry. And so we can decide how we trigger the fragmentation of those ions. Imagine that this is your compound from your plant. So we can choose an intensity that is the most common way. And then all the ions that pass that intensity, they can get the fragmentation. But there is also other ways to do it. For example, the isotopic pattern, if we are looking for compounds with sulfur or some specific compounds that they have a specific isotopic pattern, we can use that information to trigger the fragmentation. Also about neutral loss, for example, if we are looking for glycosides, we can ask the instrument to, every time I see a loss of sugar, to make fragments of that precursor. And the other one that we'll talk a little bit more is the mass effect. That is basically the exact mass with difference with the integer. I have a slide to explain that because it's a little complicated concept. And of course, for each one of these ions, we can get a peak. We can integrate the area under the peak to use it for quantification. So this is a typical chromatogram. We have the retention time. This is at 30 minutes. Now we are trying to make about 15 minutes methods. We have the intensity, and these are the two first dimensions that are more evident. But we also have the dimension of the mass charge, right? So even if some compounds, they elute together, that this is the case, because in this case, we are seeing more than 5,000 ions. This is in positive ion mode. But most of them, they separate in the mass charge, right? So they have different molecular mass. So, and at the same time, each one of these 5,000 dots that you have here, all of them has fragments. So we have structural information for each one of those compounds. So these database, these data sets are huge. And from here, we get the precursor exact mass, which we can get the elemental composition for most of them. We have the isotopic distribution, we have retention time and fragmentation information. And of course, we have the abundance of each peak. So we can compare, a treatment, let's say a knockdown against a wild type of specific plant. And so the structural information with a little bit of training and in patient, we can basically explain. If you see the name of this, the figure in the bottom, these are fragments that are coming from one of these decaffeilchemic acids. So using some rules that we know which bonds are more weak, so we can predict and which to explain each one of these fragments, right? So these are color code. For example, the 179 that is here is a caffeic acid moiety, and so on. So basically, we can, using that information, those fragments, we can put them back together and try to elucidate the structure of the molecule. And so this is a quite time-consuming process. When we start with 5,000 ions, we need to be sure that those are the good quality ions, because most of the ions that we see in the mass spec could be electronic noise, could be peaks coming from the solvent, also from each molecule. We can see like probably 100 signal for some of them. So we need to do a filtering, and for metabolomics, this is a huge part of our time extracting the good quality features. For example, we only use the features that are not in the blank, right? And that we inject a QC, that is a pool of all our samples, and only ions that they behave same, because it's the same sample. So every time you inject it, you should see the same. So whatever is higher CV than 25%, we usually don't use those. Also, we want to use for matching against libraries, we want to use only features that have fragmentation information. So from this, we can have about more than 1,000 good quality features. Then if we look for those, many of those feature will give us a lot of candidates. So usually it's very easy to get thousands of candidates, and many times we basically need to look through most of them. So for those, we look the ones that have a very nice match, and eventually we need to do a lot of manual inspection looking for insert fragmentation. For example, you can see glucose in different parts of the chromatogram, but those are fragments that are producing in the electrospray. And eventually we get to our annotations. So in this case, for this particular case of Centella asiatica water extract, we got 124 annotations. And this figure that I'm showing here is a Cyroscape network using all those features, all those annotations. And basically what we did this here was the Tanimoto algorithm, and it's putting together it's overlapping basically the 2D structure, basically the molecular formula. And so compounds that are very close, for example, here, quercetin and camphorol, the difference is a hydroxyl group, right? So they are quite neighbors. And so this make us it gave us a lot of confidence that the annotation is correct because we see them in families, not an isolated compound that who knows which pathway is coming. But when we see many compounds from the same pathway, we get a lot of confidence. So in this particular case, the one that you see with the square, those are compounds that we were able to quantify using the same analytical run. And the ones with the circle, they are only annotated. So memory in a mice. And then we run out of that plant extract. So we want to prepare another one. And so we get another from whatever the source. But sometimes that is a huge difference between using one source or another. In this case, we have 8 different plants. I mean, Centella asiatica, 8 different, but from 8 different locations. The ones that are here in the red cycle are from Oregon. We have one from India, for Ecuador, from Peru. And so they look quite different, and they have different phytochemicals. So the next step to get the quality of this extract was to quantify some of those compounds. So we quantify a family of decaffeilquinic acids. Centella asiatica is particularly rich in these compounds. A monocaffeilquinic acid, triterpenes, we have some flavonoids and some hydroxynamic acid derivatives that we have standards. This is use the standard we run in one of the methods. It was using a phenyl tricolumn that is particularly good for phenylpropanoid pathways because the phenyl tree has a phenyl group basically in the beads. So we can have pi-pi interaction between phenolic compounds and the column. So they have a very nice separation, even isomers. Of course, for this, we need to do calibration curves. This is a tough instrument, right? So we don't have the 6 orders of magnitude linear regression that we have in a triple quad, but we can still have a pretty good, sometimes like 4 orders of magnitude like this calibration curve. And so we want to validate this method. So basically we we measure the matrix effect. What we do in that case, we have the extract and we put intentionally more of the standards, and then we quantify again and see how much we got. So in these cases, we basically got between close to 100% of the recovery. Sometimes we get a little bit more, and that is because sometimes the matrix has some enhancing effects, and sometimes we have less. The most common thing is that we get less. And this is a real sample. It's a full of extract of Centella asiatica, and we can see all the compounds that we see, and we can quantify using the precursor. So for this, we use the MS1. So the instrument first do a full scan, and then it does fragmentation of the most important compounds. And then we can use the first scan for the quantification. And so we did this for this. And one of the things that I want to highlight here only is that when you are doing research to treat some cells, for example, and if you don't have controls over the production of that plant to get the extract, these things can happen. For example, imagine that your active or your bioactive in this plant is asiaticoside. So there is a plant from, I think this was from India, that it has about 20 times more of these compounds than the another plant that it grows in Peru. So imagine the variation in your experimental design. And this is one of the very reasons that, as you know, using plants as a medicine is sometimes is very controversial, right? Because it seems like sometimes work and sometimes it doesn't, on top of all the other factors. So as you know much better than I do, plants, they cannot run away when they have stress, right? So basically what they do is to produce different secondary metabolites to defend themselves, right? Or for communication sometimes. So that is the reason we never have, when we grow plants in different conditions, they produce different of these compounds. Okay, that was the first application. And so now I'm gonna talk just a little bit. That was the one that I want to put more time, because to explain how the instrument works. So this is another interesting application that we developed quite recently, and it's using spectral data and correlate with bioactivity assays to find the bioactive compounds. So this is we started by creating a chemical diversity. The first step that we do, we create a solvent partition. So we have four fractions, subfractions of the crude extract of the plant from different polarity, right? So that was the intention. And then we do subfractionation of some of those in an LH20 column, just to have 21 fractions with different diversity, let's say a different gradient of some of those phytochemicals. And then we inject those to the mass spec. But also we did a bioassay to see how the cells behave with those fractions of the plant. And I'm gonna show that in the next slide. So from this, we put together the information coming from the mass spec and also coming from the bioassay to try to find which features in the mass spec can predict what is happening in the bioassay. And from here we have two methods. One is the selectivity ratio, but the one that we worked the most was with ElasticNet. And then from here we try to annotate and have a candidate. So we try to do this much faster than doing an exhaustive fractionation, like trying to get each one of the compounds. That can take a lot of time and resources, and sometimes we end with a compound that is already known. So this is the assay that we use here. I just want to explain a little bit how it works. We have these MC65 that are neurons, and they have a gene that when we have the cells with the tetracycline, the gene is repressed and we don't, the cells can live, right? But if we remove the tetracycline, then the cells die like in 2 days, I think like 3 days. But if we remove the tetracycline and we, instead of that, put the 21 fractions, we want to see if they die or which one can have a compensation, let's say like the tetracycline. So this plot is a little bit complicated. I just want to show you, highlight some of the things. We have the fraction from 1 to 21. And this in the right, if you can see my pointer, is the cell bioactivity. The control is when we have tetracycline, right? So when we don't have tetracycline, the cells, they in 2 days, they basically die like 60%. We only get 40%. But some of those fractions, they have some of the compounds on those fractions make the cells not even survive, but sometimes even proliferate. And that is the case from this fraction. So this change in the bioactivity is correlated with the composition of each one of these fractions. So in the mass spec, we measured about I think it was 1,500 features that you see here. And then those are correlated somehow with this variability. So all these fractions basically have a gradient of many compounds, because when we elute from the column, we don't have a clean separation at all, right? So we have gradients of concentrations. So using the ElasticNet, we found that the top one candidate was the caffeinic acid. So we did this molecular network with GNPS that somebody explained in this seminar, I think, before in this biochem series. But I will explain a little bit the last part of the talk also, why we like GNPS so much. But basically here the colors are each fraction. And whenever the bioactivity was high, we know that these compounds, for example, they are in high concentration, and that is not a coincidence. Basically, it's the way that we found that in this case, the caffeinic acid were the most seems to be the most bioactive ones, and we prove that with cells also. Okay, so the next part is using mass effect analysis. Probably you are familiar with this concept. But for analytical purposes, the mass effect analysis is the difference between the integer and the decimals. So by definition, carbon has a mass of 12.000. However, those 6 protons, 6 neutrons, they have that mass only when they are in a carbon, right? If we take the protons and neutrons and put it in let's say in helium, the mass doesn't match anymore. So the predicted one with the actual mass, there is some difference. And that difference is basically what we call mass defect. And it's the reason we get the energy from the sun, and basically that all the plants that we study that they do photosynthesis, they are getting the energy from this for this reaction of converting mass into energy. So some molecules, we can use that in our advance to predict a little bit of the class of compound they are. For example, here we have ethanol, and we see the mass effect is 0.04. We have some of the caffeine, I think, is 0.08. And some compounds, like man-made compounds especially, they have a mass effect that are very high, right? So usually, natural products, they have a mass effect that is close to between 0 and 0.4, the mass effect that are coming basically from plants, but most of them closer to 0. So we use this to analyze some compounds in Tabernante iboga. That is a plant that is used in Africa for a lot of things, including diabetes. And we use the mass defect of all the alkaloids reported in Tabernante iboga. That is it's a very short range of mass. So using only the compounds following that mass defect, we got all these ions that you see here are basically, I think, we're like close to 40 compounds. In addition to we know some of these like type of alkaloids like ibogaine, we know some of the fragments like 122. So basically, if we use the mass defect in this range with looking for this fragment, we can basically be 100% sure that all these compounds are alkaloids. So in this plot, this is, I'm showing here the retention time in the X, and we have the mass charge, and the intensity of that, the abundance of that alkaloid is in the size of the sphere. And in this case, using this approach, we found more than 30 new alkaloids in this plant. And we couldn't get the standards for all of those. We got standards only for 7 of those. And all the times, the one that we predicted was the alkaloid that we found in that plant. So that is a very cool application of mass effect that works particularly good for alkaloids. So another application that I want to show is, this is a very interesting case that happened in working with coffee in Mexico. We have a group that approached to us because they saw something very interesting when they tried to do embryos from leaves from coffee. So when they induce somatic embryogenesis in Cafe Arabica, so this is a leaf that happened to be after 7 days, 14, and so on, until almost 2 months. And they don't develop embryos as Caffea canephora, that in the day 56, you can see all many embryos. And so this makes the coffee production much easier when they can do what they did here in Caffea canephora, because from one leaf, basically they can get about 100 plants. And so they basically get clones instead of having, the drift of using seeds every time. And so that was very interesting. So that means something, why is this happening? Why in one happen even though they are the same genus? So the next experiment they did was to put them together at the same time. So this is the control, this is Caffea canephora. And when they put it together, we lived from Caffea arabica. The Caffea canephora did also the embryogenesis. So this is after 2 weeks, and I think this is after 2 months. So basically, this means that the Caffea arabica is releasing something into the media that is inhibiting the embryogenesis in Caffea canephora. So we did analyze the medium of those plants to see what was releasing. And we found some of these are the most abundant compounds that we found, including chlorogenic acid, caffeine, and many others. So the next step was to put those alone and see which concentration of those compounds inhibit the production of the embryos. And eventually, we found that caffeine together with chlorogenic acid, they have a synergy that basically they inhibit that. So now they are trying to, find a way to remove them from the media and try to do, they still working with this experiment. So it was a very cool finding. So we work a lot with honey in our lab, and this is a very nice project that we want to work because basically we want to create a fingerprint of each honey, because there is a lot of fake honey in the market. If you go to basically any store in the United States or in Canada, if you get to what is the name of the store, but basically it's very high chance that you will get fraudulent honey in probably at least 40% of the cases. And so that is very bad for many reasons. One of them is because it's a big commodity, but also because beekeepers, they get their, if we take beekeepers out of business, they don't have this service to provide pollination to many, for many orchards. So, and that could be a huge problem. Not even the value of the honey, but the value of the pollination of the crops. So we are collecting, creating a big database of honey. We have more than 1,500 honey. And this is what we can do with one of these instruments. This is a timsTOF Pro that can do also ion mobility. In these cases, with this instrument, is much faster, and we can do what we call 4D metabolomics. For that, we can measure the collisional cross-section of molecules. So the additional dimension with the previous data that we saw with a regular TOF is that we don't know how big is the molecule. Basically, the instrument has a step that we blow nitrogen against the molecules, and then the drift of this, let's say the resistance of that molecule to pass through the nitrogen is correlated with the size. So in this plot, the size of the dot is the collisional cross-section, and usually it's in the order of angstroms, from about 100 angstroms to 540 in this case, that we saw the biggest molecules in the honey that are lipids. So in about 20 minutes, we can see at least 10,000 molecular features. And we use this is just, since we are talking here about plants, we try to see which of those features came from the flower that the bee visited. Because every time we see more in the stores that we have honey that they say is from raspberry, blueberry honey, sweet clover, or cranberry, or whatever. And that is a very interesting claim, because bees, they use sometimes like 5 kilometers to get the honey, right? So it's difficult to trust that. However, it could be the case that they have access to a lot of those flowers, and then they have enrichment of that particular honey, or those phytochemicals they collect from that particular flower. So basically, we did that for these different plants, and we find the main biomarkers that they can predict if it's enriched with that flower. We get the molecular features, but getting from here to the actual compound, that is still a long process to go. And so we are still working in this project. So finally, I just want to talk a little bit about why we like molecular networking so much. Basically, what we see in the mass spec is all disconnected ions, right? So, but when they are in a live organism, they are connected, right? They are in pathways, they are having communication. And this communication or this regulation of these molecules is what make a thing being alive, right? Is the reason we can feel and think. And usually what we get with the mass spec instrument is basically use dead ions, right? Elemental composition and not this interaction. So that is the reason we are very interested in doing molecular networking. So I want to ask you if anyone can guess what is this object here and this one here. Those are, let's imagine these are individual molecules. And sometimes what the mass spec see is it will not know what is it, but if we know what is around them, then we can get a better guess. So if I show you the next part, so this object, if we see that there is a bunch of persons around it and it has kind of the shape of a person, so we will know it's a person because the neighbors around. In this case, if we see this object here and we see that it's a bunch of rocks everywhere, so we know it's a rock. And that is because we know what is around them. So that is what basically NPS can do. And so we can put together this to get a lot of information. One of the things that we work in the past is that, with mice, it's very easy to force them to eat some plant and let's see what compounds they get from this particular plant. But with people, if you give them the diet and they do it at home, many times they forget, right? And then that wastes a lot of resources in the experiments. So one of the experiments that we did was to do a small test, and with lab colleagues in the lab, we eat a different food, each one of us, and then we analyze urine. So, and then we do the ENPS networking, and we see, for example, just to put an example here, this compound that is called abenasterol is only in one person that basically enriched in one person. And the person who eat, in this case, oats, it get that compound. So we can see colors that are biomarkers. The one that is only in one of the persons, that could be a good biomarker to know if that person eat the food or not. And we also, this is very interesting for if you want to see the effect of a particular diet in, a plant or a natural product, let's say a extract of a plant that they claim is good for memory or whatever, then we can be sure if they take it or not. Another thing is the propagation of annotations. For example, in this work, we have only one standard that was xanthohumol, but we were able to annotate many of the compounds around them because basically the difference is a methoxy group, a hydroxy group, and then we can propagate. And so to be together, then they need to share at least 7 fragments. It's a threshold that we can choose. And so those molecules have at least 70% of the fragmentation matching. So this helps to increase the confidence by a lot, because the noise will never have, it will never share 7 fragments with our standard. And we are doing this also to finding some hormones in some organisms. And basically, yeah, those are the applications. I hope we have a little bit of time for questions. So I just want to acknowledge this fantastic group. Is the best lab that I've been by far, and also with the Professor Leonard Foster. And also I got a lot of fun. It was a very nice place to work with some experimenters with planned with Professor Claudia Meyer and Fred Stevens. And yeah, I like to grow chilies. All these chilies here are, I grow them in the summertime when possible. So I'm not working with plants too much, but I'm a big fan of plants.
Thank you very much, Armando. Questions for Armando, then? We have plenty of time for a discussion here.
I have a couple of questions. Oh, sorry, I see one in the chat.
I think go ahead, Eric, and then
Yeah.
Federico can go.
Okay, so two quick ones. The first one is regarding the honey. Do you have a reference to know where what should be the compounds that honey coming from a particular plant really has? Because I suspect you can prevent honey and bees from going from one plant to another, right?
Yeah, that's exactly the point of this project. So what it happened many times is that if you put the hive inside of a farm, let's say blueberry farm, the bee is gonna find the flowers close by, so they don't need to travel. So, but it's still they can do that, but probably more than 90% of the time they are gonna use the blueberry. So that could be the case that we trust those, and we do to put them in the model to predict the flowers that the honey that we don't know. We need to use honey that we trust 100%, right? And so that could be done only with local markets and collaborators, not with buying the honey from a store. And another thing that we are doing now, I mean, part of the team is doing is collecting nectar from the flowers also then to match. But many times it's not that simple, because once the bee eat the nectar, then it has a big transformation. So you can lose glycosides or put more glycosides. So you can, your marker can be a transformation of the compound that was in the flower. But that's a very good point. And that's exactly the idea of this project, to have a database and see what we can trust.
And the other is a quick question that I'm sure many of us are interested in. That collection of 450 compounds, is that accessible? I mean, you guys build that and it's kind of for your own consumption, because I think many of us would actually love to have 450 compounds, access to 450 compounds.
Yeah, that is a library that we acquired. Basically, we use token aliquot of the library, and we still have some of the, because we still have some of the, let's say, the pure compound, because metabolomics, we have so many methods. You know, you see, depending on the class of molecules that you want to see, it's not like proteomics that you basically use almost one method. So we see some compounds in positive mode, some compounds in negative mode. Sometimes we want to use helic, a C18, phenyl tricolumn. And so we have built the library for a method that is mostly useful for plants, for a secondary metabolism, and we run those, and those are available if anyone wants to use it. And yeah, but if you are interested in a particular compound that is that we need another method, we can use that standard again.
Thank you.
Thank you for the question.
I think the next question here is from Federico Roda.
Hi, Armando. Very nice talk. Very informative. I wanted to ask you a question. So when we work with plants, most of the compounds cannot be annotated with these methods. And as the last question suggested, there is not so much data sharing or resources to actually use these computational methods, or to take the most of these computational methods to identify really new classes of compounds or things. And so do you see that this is changing? Because I come from the genomics world, and well, maybe genomics is easier than metabolomics, but there is much more of a framework, and you can have an idea of most of the genes, what they could probably do. For metabolomics, that actually doesn't exist and is normally dealt within very small communities, and even with the GNPS and all these things, you can annotate a very small proportion of the compounds, and normally those are the compounds that are in most plants, flavonoids. But the ones that maybe are the most interesting, we don't know anything about them.
No, that's a very good question, and it's a big problem. So from now on, we are trying to put the data that we will acquire, even raw data and processed data, in the metabolomics workbench. So in the future, hopefully, when more standards are available or new methods, or hopefully artificial intelligence will help us to annotate more compounds, that data is available. But I know I feel your concern, and it's something that we struggle all the time. That some of these compounds, if you want to buy a standard, sometimes it's $500 for 1 milligram. So imagine that, as you say, we annotate like 5% of the compounds of a plant extract every time we analyze them. So we really, and this has been a topic in the ASMS and other conference that we want to share more, and we should do it. And especially, when you see libraries that they were creating using taxpayer money, and then you need to pay it to get access to those libraries. I feel like it's really unfair. So yeah, I'm pretty up. I think it should be almost mandatory to put those available when you do a publication. Sometimes you have, sorry, yeah.
No, go on, please.
Yeah, sometimes it could be the case that you are applying for a patent or something like that. Not our case, but it could be that you want to protect your finding, and so that could be the case, and we probably need to understand that. But most of the compounds are just exploring the plant, so we should share those.
And it's a huge hurdle to use artificial intelligence or these methods that require actually a data set to train the things. If you don't have shared data sets, only the data sets that you get from your own lab, it's very difficult. Well, thank you very much.
Thank you for your question.
I think the next question was from Lana, but perhaps that question has been answered now.
I have a question. I was curious if I don't understand the mass defect completely, and I don't think I will, but I just wanted a quick clarification of, was that you modified the samples, or was that looking for a specific defect, or a specific feature in the chromatogram?
Yeah, so we didn't did any derivatization or transformation of those samples. Basically, in that case, for alkaloids, is quite straightforward, I will say, because alkaloids have this nitrogen. So nitrogens love to get a proton in electrospray in positive ionization mode. So you are very easy to see them in very low concentration. So what we did for this plant that we know is very rich in alkaloids. Actually, now is legal in the United States because so many alkaloids. So for all those reported in the literature, basically we saw the mass defect of that range of compounds. It was a very narrow range in the mass defect. And then we applied that filter and asked to the mass spec to basically see only those compounds following that mass defect. And that allows us to see many, many alkaloids that also for further confirmation, we want to look for a fragment that is typical for this class of alkaloids. So if we have the mass defect and we know a fragment that is coming from an alkaloid, the retention time match, the ionization mode match, so it's very likely that those are. Furthermore, we only put alkaloid reported in other plants. We didn't put any new alkaloid, just to be sure, but who knows how many new ones are.
Okay, so that would be similar to, like, if you're applying a filter to the mass spec, would that be similar to like looking for a specific isotope ratio?
Yeah, it's quite the same. You can, to trigger the acquisition, especially for the MSMS, you can use different things. And one of those is like the isotopic pattern, as you mentioned. It could be the mass effect, or it could be a neutral loss. So you see that you lost a sugar, then ask the mass effect to do a fragmentation. Or it could be a threshold, that is the most common time, that whatever have some intensity to have good quality MSMS, then it triggers the fragmentation.
Okay, thank you so much.
Thank you.
I think the next person to raise their hand was Gaurav and then Ankur.
Hi, thanks for the nice talk. And my question was similar to what Federico and Eric asked about resources, and especially with in terms of identification, concern is of misidentifications from these spectral matches. Right now we are looking at GNPS, something that GNPS identified as a capsaicin, but the sample is from a gymnosperm. So it certainly doesn't exist there. And the issue then is, how can we, so commercial software to include multiple evidences sometimes, like Progenesis has multiple evidences to provide confidence in a specific identification. Is there a workflow that's open source that others can use, which is not commercial, that enables such a like assignment of confidence in identifications?
Yeah, that's a bottleneck now of metabolomics, right? So there are some software that are trying like Sirius and MS Dial, and they are quite good, but it's the same problem always that you get what we call level 2 or level 3 confidence in the annotation. So, we have different levels of confidence. And that is one of the reasons that we wanted to have an in-house library, because for those compounds, we can be 100% sure. But everything else, we cannot, if we don't have the standard and we're running in the identical conditions, using the same collisional energy and using the same gradient, the same column, the same HPLC, because sometimes even the dead volume is different in another HPLC, do you have different retention time? So you lose one of the most important parameters, that is retention time. So then you always need to call it a level 2, no matter how sure you are. And you say, this is a plant that is called Centella asiatica. This compound is called asiaricoside. So that makes sense. We still cannot call it level 1 if we didn't run the standard. So yeah, and there is no easy way around that. Like 10 years ago, when we started with machine learning, we thought that machine learning will fix all that for us. And it didn't happen at all. So now we hope that artificial intelligence, but who knows, hopefully. But it is, I think the safest way to do until now is to say that those are level 2 or level 3. Yeah, that's a very important point.
Already we have 2 to 3% being identified, and then if we trim off even the level 2, level 3, then it's like less than 1% that we can actually use from this data. And so I think identification-free methods like PCA or the information theory-based methods or something else that can enable more insights from just the metabolomics data, molecular networking. I think those would be pretty good to standardize across the field.
Yeah, it still has some very good use, because I think if you have a level 2 annotation, and that is your compound, that it will decide if you is a fake or authentic plant, then you want to synthesize it or by the standard, and then you can validate. So if we only use our in-house libraries, we will find all the times compound that everybody knows, right? That everybody did. So to expand that, we need to get these tentative annotations and then try to explore more and synthesize them, or do knockout pathways and see where is that compound coming from and what it's doing.
Thank you.
Thank you.
I think the next question here is from Ankur. It's in the chat. I can read it, or Ankur, if you prefer to ask it yourself, that would be fine too. I'll go ahead and read it. It says, "Hi, such a wonderful talk. I want to ask one question. Did you compare your metabolomic result with another method like NMR metabolomics?"
We did it for the Centella asiatica project, actually. We were part of analytical core that is supposed, from the NIH, that is supposed to study natural products. And so I don't have expertise at all in the NMR. But that's a good point, because sometimes some of the standards that we bought from some companies that I don't want to say the name because it's getting recorded, but some of them, they were not the isomer that they say. And it happened too many times. So we couldn't trust that. For example, if we buy from 2 different sources, they have different compounds with the same name, especially with the caffeinic acids, because 1,3 and 3,5, it really starts sometimes. How do you count it? And it's so much controversy. Some of those compounds has the same name. So at the end, every time we get a new standard from a new company, we did NMR. Somebody in the group did it, and then it say, okay, it seems to be the right one, or it's not. So then we will change the name, right? Because we know which one is it with the NMR.
Other questions for Armando before we are one step closer to the weekend? Okay, well, thank you, everyone.
Yes, sorry, just want to say that Happy International Women's Day. Yeah, thank you for the invitation too.
Yeah, thank you, Armando. Well, we will hang out for a few more minutes in case there are any other questions for Armando, but otherwise, thank you everyone for being here today, and we will see you in a couple of weeks.